with that, I want to introduce you to Dr. Andrew Award. So the title of his talk is Speeding Up Vaccine Design and Antibody Discovery Using cryoEM and Machine Learning. So Andrew received a biology degree from Duke University and a PhD biophysics from the Scripps Institute, where he is currently. He and his lab, they have made outstanding contributions to the antibody biology field, and his lab's work have pioneered the study of serum antibodies on the molecular level. Okay, not only that, but also combining that with cryoEM studies on antibodies discovered from the B-cell receptor as well. And today he's going to tell us about exciting new stuff from his lab. And Andrew, we are very excited to have you today and welcome.
Thank you, Camila, for the nice introduction. I will pull up my slides here. Does that look good?
It's perfect, Andrew. Thank you.
All right. Okay. So good morning, everyone, or good evening, depending on where you're joining from. Today, I'm going to walk you through, really what has been my lab's effort across a lot of different infectious disease space, primarily around developing tools that can really accelerate both, vaccine design as well as antibody discovery, and using cryo-EM and computational methods, which have really, grown in their power over the last, 5 to 10 years. So, So when I started in the field, really the state of the art in a way for viruses or envelope viruses was to stabilize, figure out a way to stabilize the pre-fusion form of the primary, antigenic, immunogenic target on the surface of viruses. And so work by Jason McClellan and colleagues, on RSVF, which is now, encoded in vaccines. Work we did with John Moore, Rahir Sanders, and Ian Wilson, provided the first attempts to do this. And then, these attempts to stabilize prefusion have been scaled to many other envelope glycoproteins, including the SARS-CoV-2 spike. And really the idea here is, in RSVF, for example, if you just make a virus, most of it is in the post-fusion conformation. If you make antibodies to the post-fusion conformation, they're not useful. So by stabilizing these metastable proteins, you have a better chance of on-target responses. We've since evolved quite a bit, and particularly for HIV, influenza, others that are variable and change. We're looking at more precision approaches. So trying to eliminate off-target responses. These are very large surfaces. So there's on-target and off-target encoded on the surface. And so we can try to do epitope focusing or epitope scaffolding, where you just take, for example, the CD4 binding site on a mini protein and present that to the immune system. And then germline targeting is sort of the ultimate version of this, where you're trying to design a protein that turns on a specific VH or VL lineage that we know from human infection primarily can encode a broadly neutralizing antibody. And so this has actually been very successful. And my colleague, Bill Schief, has published quite extensively on this. But in a human clinical trial using this germline targeting approach of encoding just the CD4 binding site epitope, they were able to turn on specific VH1/2 and expand those, and basically every human that had the actual allele. So that's the level of precision that we're trying to achieve for HIV and now for many other viruses as well. But it takes a lot of work to iterate and get to that point where you have that resolution, if you will, and confidence that the protein designs you're making are eliciting the responses you want. So as I mentioned, this is an iterative process, and it starts usually from an immune individual or infection model, We look at the antibody B cell repertoire, and that's now increasingly done by sequencing and sorting of antigen-specific B cells. And then we do a lot of, my lab in particular does a lot of structural analysis to understand and kind of map back where those B cells are binding. That can go into a vaccine design program. I'm not going to mention it today, but all the new tools for AI-based protein design have taken over, and this process, which used to be quite arduous and time-consuming and involve lots of rounds of Rosetta and mammalian display and other things, can now be done quite rapidly through these modern protein design tools. But again, you only know what to design once you understand what that design elicits. And so a lot of these things that we're doing are also changing the way even clinical trials are designed, right? So the endpoints for clinical trials aren't necessarily, is this a neutralizing or efficacious response? It's, is this on target at the sequence, molecular, and structural level? Um, and so, again, we need really high resolution, high quality tools to evaluate how well we're doing, particularly as we get to outbred or heterogeneous model systems or humans. And so I really like this graphical abstract that was put together by Tom Canyles and Rahir Sanders' lab, and Gabe, who's an institute investigator with me. And really, here again, this is a germline targeting approach. And the readouts were the epitope-specific B cells, as well as a lot of structure determination to show that they had achieved in about 75% of the patients what they intended to do at the molecular level. And here's just, a gallery, right? What used to take us, months and months of time to solve cryo-EM structures of now takes a matter of days. And based on this clinical trial where many, many different antibodies or B cells were isolated, Gabe and the team was able to solve many structures, again, understanding at a molecular level, can these antibodies bind in the same way as a known broadly neutralizing antibody? Can they navigate, in particular, this one glycan hurdle that HIV uses very effectively to mask the CD4 binding site? And so, again, by solving structures, we have a lot of confidence that this vaccine approach is going in the right direction. It's priming the right responses. And then, here's a clever way that Gabe came up with to kind of look at and compare, right? So structures are complicated, three-dimensional milieus. Here's kind of collapsing it into these plots that maybe make it a little bit more easy to digest. So here's broadly neutralizing antibodies we know from patients that can neutralize, 60, 70, 90% of viral isolates. These are the antibodies we're trying to achieve through vaccination. If we look at, what they have in common, right? They all, in this case, use HCDR3 on the antibody to primarily interact with these key conserved residues. And then, of course, they also use, in this case, all their light chain loops to interact with this one key glycan. So you can start to see a signature of how broadly neutralized antibodies converge on this phenotype very effectively. And now, if we compare that to some of the early vaccine elicited antibody responses in humans now, what's nice to see is that as designed, the CDRH3 is interacting with similar residues, but you can see that some of the links to other areas are a little bit weaker, less conserved. There's a lot of additional and off-target, if you will, interactions. And that you can see the CDRs actually interact for the light chain interact with a lot of different residues. So the idea now is, okay, so you've primed a response that looks good. How do you boost or, shepherd the response and evolve the antibodies with booster designs to get a phenotype that looks like this? So we can use these metrics now to evaluate how well we're doing. Okay, so what I just sort of talked about was really the field standard. You take one monoclonal antibody, one antigen, you solve a structure, you analyze it, and then you do that, in stepwise. And that's kind of where we were when I started the lab is, waiting for people to isolate antibodies many weeks after immunization. And then we would do our structural analysis, and so that you could see the feedback loop for structure was quite delayed. Um, but with the onset of a lot of new technologies, so I mentioned the high throughput, B cell screening, people isolating B cells from, after one or two immunizations. 10x and repertoire sequencing have given us a lot more information, you know, really early on. And then our work, which I'll describe, MPEM, provides a structural look at what those responses are doing. So these have really netted into a lot of high content information very early on. So that iteration loop is quite a bit faster and a lot more depth of resolution. So, just providing some references here, this has really expanded quite a bit, and people using DNA barcoded approaches like LibreSeq from the Georgiev lab is uh, really, given us a lot of high content information. Mass spec from George Giorgio and Albert Hecht's lab and others is giving some sort of proteomics level information, and then MPEM, which is the structural aspect, and I'll spend more time talking about this. Okay, so NPEM sort of started with, you can run an ELISA, and ELISA gives you sort of this monotonic curve that recapitulates just a bunch of stuff binding. So the idea was, can we put this in an electron microscope and deconvolute the heterogeneity using the power of single particle, two-dimensional and three-dimensional classification? And so you can see here a nice composite map, right? It's in black and white, but because we know something about the underlying structure, we can start to assign color based on epitope. And then you get some information on, again, what's immunogenic, and then you can look at what's immunodominant and add some, quantitation to it as well. So in addition to getting a nice readout on what's being targeted, we've started to see some new themes that maybe some expected, like that you have a epitope hierarchy and immunodominant epitopes, and some of them saturate early on, like you can see these purple ones saturate early on, and then only after time and boosting do you get these, on target, if you will. We're starting to see more in terms of antibody feedback, antibody-induced destruction. I'll show an example, higher order immune complexes, and even autoantibodies. And I'll allude to some of these features. But all of this, right, it can be happening on an ELISA plate, but you can't really tease apart the mechanism. But when you have the visual structure, you can start to do that. So this is our graphical abstract. Hannah, Matteo, and Lars and I published in 2018. We started with rabbit sera from an HIV antigen immunization. We did our epitope mapping, and it worked beautifully after a lot of troubleshooting, of course. And we even did some cryoEM, which indicated that we could go beyond the sort of blobby low resolution, and that'll come into play a little bit later in the talk. And again, as I mentioned now, right, when clinical trials are looking at molecular phenotypes, it's also nice to have a readout of what patients are doing. And so now this is MPEM data from the C101 trial with that germline targeting HIV antigen. And you can see this is after two immunizations at week 10. You can see, we see a lot of patients that have these base antibodies, some that bind this glycan hole. We have actually some placebo in here. This is blinded, but we can guess that these are placebo based on no responses, and then sporadically some interesting responses. But after three immunizations, now you can see that the epitopes diversify quite a bit. So these immunodominant epitopes are saturated. The immune system starts sampling these more cryptic or difficult to target epitopes. And most importantly, 72% of patients showed responses against the CD4 binding site, which was the design principles in which this GT, this germline targeting antigen, was made. So that was very encouraging to see that, and also provides information on which patients you're most likely to pull the B cells out of that will have these CD4 binding site reactive cells. So these are now, particularly for HIV, but for other clinical trials, these type of analyses are fairly routine for us as readouts, and to inform and really complement a lot of the other serological approaches that are being undertaken. And I will say, if you notice, over time, you start to see this protomer phenotype. So that means instead of a trimer, we start to see monomers bound to an antibody. And so we sort of presaged this several years back. And this is, again, beautiful work from Hannah, where when she was doing MPEM, we noticed after the priming response, we got our typical trimer plus, one or two antibodies, and that looked good. But then after multiple boostings, we started to see these protomers, so monomers bound to antibodies. So despite this being, a very highly thermostable and designed antigen, it was falling apart in vivo. And in fact, the antibodies were the things that were causing it to fall apart. And so um, what you can appreciate is that you have sort of the neutralizing face of the antigen, and then anything internal is an irrelevant or off-target epitope. So if you are degrading your antigen in vivo, you're going to get responses to the neutralizing face as well as the non-neutralizing face. And this is not just happening in HIV envelope, but a lot of other antigens is antibodies can bind to certain epitopes, unlock and cause them to fall apart. In the case of HIV, this was really informative data, and we actually then, Chris Cottrell actually designed a disulfide bond that prevented this phenotype. So again, on an ELISA plate, this and this are identical. You wouldn't be able to appreciate the difference, but MPEM really showed that this was a potential liability and a design opportunity. Okay, so now I'm going to switch and talk a little bit about Lassa GPC, which is another viral glycoprotein target that we work on. Work from Philip, Alex, and colleagues in Niels King's lab really set out to stabilize what is otherwise a really unstable glycoprotein, Lassa GPC. They achieved this through some protein engineering of the GPC, as well as adding one of these I5350 trimeric scaffolds designed in Neil King's lab, which really is a scaffold that keeps the trimer together. So it had two advantages. One, it stabilized it like a protein, and two, it could be self-assembled with a second component into a nanoparticle, which is of course a nice vaccine modality. So Haley, a student in the lab, former student in the lab, took this and said, okay, let's see if we can not just make Lassa GPC from the Josiah strain, but many other strains as well. And she achieved that in her PhD. And again, using the scaffold and protein design technology. And now we had a panel of Lassa GPCs to use as either B cell probes, structure probes, serological probes. And we said, okay, that's cool. Now let's try to use this, our MPEM technology, to understand immune responses to Lassa GPC nanoparticles in rabbits. So we took the trimer, added our polyclonal Fabs, and lo and behold, every observation we saw was these monomeric Lassa GPC. So the antibodies binding caused this to fall apart. And so here's just some at cryo-EM resolution. I'm just showing you, in all the rabbits, you can see multiple antibodies stuck to these individual protomers. And you can see, again, just like HIV, you have the intact surface of the trimer, the neutralizing face, and then here is the monomer. This is the non-neutralizing face. And so the majority of antibodies elicited in the rabbit model were against these non-neutralizing epitopes. So off target, distracting, and really running counter to a productive immune response. We're still, I will say, Lassa GPC still remains a very difficult design problem, and we and others are trying to fix this. But Philip came up with a nice workaround, right? We still wanted to understand what was happening on the outside neutralizing face. So what he did is he pre-complexed Lassa GPC with known antibodies, one that binds to the apex and kind of holds the trimer together, and one that binds near the base and again holds protomers together. Then incubated with the polyclonal Fabs, and then did our MPEM pipeline. Um, and so this actually worked really well. We didn't get protomers because we're using those stabilizing antibodies. And you can see now in all of the rabbits that have a neutralizing response, right? And these are neutralizing titers. They make blue antibodies. And then all the rest make, these orange and red antibodies. And so you could also see the progression of that over time. And so that gives you already a low resolution correlate that the blue antibodies likely are the reason why you have neutralization. We of course want to know, what's happening at high resolution. Um, and so again, we can now, instead of doing low resolution negative stain, we do our cryo-MPEM experiment. Um, and just to say, this is really nice because from, a drop of blood, we're getting high resolution information, and it doesn't require B cell isolation, expression, random mutagenesis, so on and so forth, which is expensive. And in an under-resourced field like Lassa, you just can't do those assays. So this is one of the rabbits. This is the cryo-MPEM structure. You can see really high resolution details. If we zoom in, we know, of course, what the sequence of the Lassa GPC is, and including the glycans. We can build that in. We don't know what the sequence of the rabbit antibody is, but based on the density map, we can build at least carbon alpha models of what the CDR loops look like. And then here's just the density map again, just to show you the quality of the fit. And again, we don't know what the underlying sequence is, but we can see that there's two bulky residues that are juxtaposed against a couple of acidic residues here. And so we can make the hypothesis that if we mutate these two residues or reverse charge, then we're likely to knock out the antibody response. And that's what we do, right? So we now, by just making two amino acids based on that structural information, we knock out the neutralizing response. And again, reconfirm that this is the correlate of neutralization in this animal model. What was interesting, right? We had two rabbits that made responses. We did our high resolution work, again, able to model it at fairly, high confidence. And what was really interesting to see is, right, in two different rabbits, they made convergent responses on this neutralizing epitope, and so much so that the CDRH3 overlapped almost perfectly, but you can see there's differences elsewhere. So these are likely different antibodies, but they have the same CDRH3 going into the same pocket, interacting with the same residues. Um, and just to say that both of these overlap highly with this GPCA1 antibody that's known to block LAMP1 receptor recognition. So it all fits together. And again, we didn't have to do a big B cell isolation campaign, monoclonal, expression, and so on and so forth. So we got a really nice detailed story from using this MPEM approach. Okay. I mentioned, we started to see interesting and new things. One of the things that Sheridan found, and we're finding, in different places, are these higher order complexes. So again, right on an ELISA plate, you'd never see this, but here we see the HIV antigen. We see these antibodies that interact with the antigen, and then we see these other antibodies that only interact with the immune complex and touch the primary antibodies. So we don't know, why or how, or if these are good or bad, right? They could be autoantigens, but they could also help potentiate ADCC or other, cell-mediated phenotypes on an infected cell, just by adding shear mass to it. So we really want to know what these are. Are they good or bad? Are they cross-reactive? the challenge is we don't have these antibodies, right? We don't have the antibodies that have been isolated, and even in a B cell antigen specific sorting campaign, you'd never pull these out, right? Because you're primarily just using this antigen as your probe to do the B cell sorting and sequencing. So it really is the sort of impetus for us being able to go directly from these type of images into making monoclonal antibodies without, having the additional information from the B cell sorts. And just again showing really, the quality of resolution where you can see the bumps which correspond to side chains. So it's quite tantalizing in terms of being able to assign the structure directly from this very heterogeneous polyclonal context. And that's really been the basis of what we're pushing for. Um, and really to kind of empower, the future, right? We want to be able to do more um, quantitation using MPEM and mass spec, as I mentioned, right? So you get a proteomics level view of what's going on to compare to the all the cellular sequencing that's done. As I just mentioned last slide, we want to predict the antibody structure with high fidelity from the cryo-EM map. And there's an opportunity to solve tens of thousands of these structures to really fill in gaps in AI. And then this will lead to being able to predict structures of antibodies from sequence alone, predicting epitopes of antibodies from sequence alone, and then being able to basically design, antibodies with atomic accuracy purely from sequence, right? So these are all kind of these tools and data campaigns to build towards these more autonomous and enabled AI methods. And so in order to do this, we need thousands and thousands. And of course, the applications are far beyond infectious disease. You can imagine for autoimmunity, allergy, cancer, and so on. Okay, so I'm gonna just talk briefly. There is a lot of effort in my lab and others to predict the epitopes of antibodies from sequence alone. Nick Wu, who was formerly at Scripps, published a really nice paper in Immunity describing this. And this model works really well for hemagglutinin, influenza hemagglutinin, and stem-based antibodies. And it works well because there's a lot of information in about those antibodies and VH169 responses, so on and so forth. But it doesn't scale, across novel antigens and novel epitopes and things. So it really shows that there's opportunity here, but there's a data limitation, and that's again something we're trying to address. Okay, so in order to address that, again, predicting the sequence of an antibody from the structure, and then moving towards solving, tens of thousands of structures. So in order to do this, Alex and team really developed and translated what was low resolution imaging into cryo-EM imaging, a lot of computational classification, and then combining, right, these density maps where you can get some idea of the amino acid residues with next-gen sequencing libraries of antibodies to compare and kind of fit in. And so, the analogy I like to make here is we're sorting immune complexes computationally, right? So each one of these little darker gray dots is an immune complex. It's an antigen plus one or more antibodies. Um, Through all of these really nested and complicated masking and classification steps, we can zoom in to a single epitope and start to dissect out the antibodies that bind there. And it's just like a flow experiment, right? Where you're gating on different features. Here could be trimer versus monomer. Does it have an antibody? Does it not have an antibody? Is there an antibody bound to epitope A? Take all of those single particles, get a reconstruction, and then with proper resolution, we can start mapping to these, very dense tens of thousands of sequence um, plots to look at all the sort of clonotypes in there. And it's pretty sensitive, and we can go down to about 1% of the data to pull out and extract antibodies. Um, and we can actually distinguish, two different antibodies that come in at a slightly different angle of approach to the same epitope. So this has become really sensitive. Alex, we published on a couple of years ago, showed that you could do this. So he could infer the sequence directly from the structure and using some next gen libraries and made antibodies that looked exactly like the polyclonal maps. So we've skipped the B cell isolation step here. Okay, so when Alex did this, this was a tour de force. I needed a couple examples and it took months and it was not easy, right? Um, Now here's sort of an interesting, case, right? And it's sort of, when you have success, you create new problems. So this is a nice experiment from Shu and Gabe, where again, showing the middle of a low resolution map, right? We see roughly 4 epitopes. We take that into cryo-EM. This is a single time point from a non-human primate that's been immunized several times with the HIV envelope. And now you can see up to 20 cryo-EM maps at high resolution. You start to get some quantitative information, right, about some of the immunodominant epitopes, the blue and purple here. Um, and the resolutions are generally good enough to start to assign. The problem becomes, this is years of work almost for someone to go through and interpret all these maps. And so it's very difficult. But thanks to James, this has become a lot easier. James and a new tool called Model Angelo. So this recently appeared in Science Advances. And so James took that original cryo-EM map from Alex, and instead of months of work, used this AI-based tool from Short Shares to start to assign at least fragments of this antibody sequence, right? Because remember, we know the sequence of HIV envelope. We can build that and dock that in no problem. The antibody is unknown. So Model Angelo is an automated tool that fills in these, short contigs, and then you have a more automated way to, or at least automated starting point. I won't go in detail, but just to say that James had to spend some time hacking this, right? Because what Modelangelo outputs is really a logo plot of probabilities, right? And so because you have ambiguity, noise in your data, resolution limitations, so on and so forth, you might not be able to disambiguate the difference between a tyrosine, a tryptophan, and a phenylalanine. Um, and a lot of antibodies, we know enough about antibodies to know that a lot of the frameworks are conserved. So and you can use different um, tools in the hidden Markov model, as well as tools like ablang to kind of really reduce the sequence space down from this highly ambiguous to get something that looks a lot more like an antibody, right? So Modelangelo works really well if you know the ground truth of a sequence, less well when you don't know, and James added these layers to improve that. Okay. So now instead of, Alex ordering 30 or 40 antibodies or guesses, we're down to a much higher success rate for many cases where the resolution is high. And this is really useful. So James used this as a way, and really the proof of concept and punchline of the paper was, can we use this as an engine to discover functional antibodies, in this case against influenza neuraminidase. So in a mouse immunization, we drew sera. You can see that the neuraminidase in the middle here is just covered in antibodies. We know from a lot of prior work that the best antibodies bind to the sialic acid binding site, so the functional catalytic site of the protein. So we could dispense with most of the responses and zoom in particularly on the catalytic site, get quite high resolution information, use James's Modelangelo modified approach, as well as some mouse immunogenetics to kind of again constrain the number of options, from millions down to a tractable number. And then he tested and tried a lot of modern antibody design tools. So once we had an antibody starting point, you can use Rosetta design, protein language models. We layered in some molecular dynamics, and then we're currently doing some conventional library display approaches to improve these even more, right? So this just provides a starting point for you to either computationally design or display, select for even better versions of that antibody as a starting point. Um, just to say, a lot of these tools also are good at mutating outside the CDRs. And what we found is, if you use these tools, you can actually improve the expression or stability of antibodies, which is, part of the battle when you're making guesses. If your guesses don't form stably, then they don't express well and you can't test them. So these tools can be really good at that. Okay, so James was quite successful, and we got 5 different unique solutions to the active site of neuraminidase. You can see with different VHVL usage, different CDRs, and James was able to validate each one of these with a high resolution structure. Um, and so really the sort of overarching, aims were achieved, and I think actually more successfully than maybe we imagined because we had now 5 different interesting starting points. They each have their own angle of approach because they have really their each own VHVL that they've been scaffolded on. They have a range of affinities. And what was interesting in particular is, 3 of them overlapped really highly in the active site, and 2 were offset a little bit. So we still don't fully understand and formally understand why some of the molecular contacts are conserved, some are not. But the combination of providing a strong structural constraint with our cryo-EM maps, the use of Model Angelo and other, AI machine learning methods converged on these 5 solutions that expressed well and also bound the active site. So it really shows the power of combining all these AI tools with a strong constraint. And I think that's a really nice example of where we're going and providing many more constraints structurally to empower sequence-based AI methods, language models. Okay, so really the real test was, are these, protective in vivo? And indeed, in collaboration with Grazi and Florian's lab, we showed that they protected to different levels, but one GO1 is sort of a field standard from Ali El-Abedi's lab, a broadly neutralizing neuraminidase active site antibody, and it's actually 100% protective. And you can see that we had ranges of protections, but one of them was equally protective, at this dose, 5 mg per kg, as the control antibody. So again, a pretty nice success story of going from an immunization to a monoclonal antibody, cutting out a lot of the steps in between. Um, and so now we're trying to combine all of the structures, binding data, functional data, in vivo efficacy. And so we have lots of data now to understand what the molecular constraints are on the active site and how to maybe design better antibodies in the future. Okay, so as I've been alluding to, right, this is the sort of current pipeline that many people use quite successfully, right? You take an immune model, a humanized mouse or a human, you enrich for the B cells. Oftentimes you're doing an antigen-specific sort to only isolate B cells that interact with your antigen. You do cloning and you make, hundreds to thousands of these, maybe 100 express. And ultimately, what you're going for here is to characterize them, their binding, their biophysical properties, and then ultimately the structure, because the structure allows you to really rationally design, optimize, create libraries. And so what I've shown in that previous example is, right, we can go straight from immune sera to the structure and kind of shortcut a lot of these steps. And these steps are still important, but it also is useful for informing, like if you have 10 different patients and we can provide the structures, then you say maybe these 3 patients are the best ones to sort from because they have the most interesting antibody responses. Okay, so just to kind of in the closing minutes here, we're really trying to extend and expand this technology. Alex, before he left, did a really nice study on an enterovirus, so a capsid virus, and showed that you could do MPEM not just on a small antigen, but on an intact virion with high symmetry. And what was nice about this study is, Alex found antibody responses to interfaces between different subunits, and you'd really only find that if you're looking on the intact virus. And we, of course, are also quite interested in applying not just a single particle, but to cryo-electron tomography of more variable viruses like envelope viruses. Um, the examples I showed previously were all on relatively large antigens, and cryo-EM historically has had some size and sort of feature constraints, but we're now, we and many others in the field are working to make this applicable to much smaller antigens. Um, and here's just, a couple examples from an HIV scaffold and malaria that show, we can go quite small now, which is good. I alluded to, adding more information, right? A static structure is good, but one that's hydrated with waters and moving is actually even more informative. So Monica and Joe have really taken this on and started including these information, beyond what we do at the simple peptide level. Um, Lee and Alba um, recently published a paper and showed that you could miniaturize and automate this. So this MPEM would take one to a few milliliters of sera, and it would take days of hands-on um, intervention. With this new device, we can get down to 20 microliters and in a couple hours be doing all of the same workflow. So this really helps us get through a lot more samples, and there's a lot of other advantages. We're quite interested in multiplexing, right? So can you do not just one hemagglutinin at a time, but four hemagglutinins or more? You can imagine in an EM situation, this hemagglutinin looks like H1, H3, they all look the same, but if you put a protein tag on it, like the superfolder GFP from Rob DeVries, you can differentiate. So that gives you sort of like a visual barcode. And then Renee, who has a lab, her own lab now in China, is working on DNA barcoding technology with DNA origami to array different antigens. And again, that gives you information. So antigens that are very similar can be encoded in different places and act as readouts. And then I think, really one of the unsung heroes, or what happens in the background, right? In order to do a lot of the work I just mentioned, um, it requires a really robust database and software development to automate aspects. And so Daniel and Charlie have really done an amazing job of propping up this database and pipeline, and Nick in the lab has really built out this database, and it's forcing people to upload and interact with and get all of the data and metadata in a useful place, because that's what you need to do if you want to actually train and do machine learning. So this is our lightweight structural antibody omics pipeline, as I like to call it, right? Now, especially with the miniaturization, we can go into rarer samples, biobanks, and our goal is to access lots of different immune repertoires with lots of different antigens or diseases, right? Because most of the information we have, at least structurally and even sequence-wise, is focused on HIV, influenza, a couple cancer, targets. But in order to kind of make these tools and enable the machine learning approaches more broadly, we need diversity. So that's kind of one of the threads that we're really working on. I mentioned this device, in terms of getting through the number of samples. This is going to be essential for us to actually do this work, and then the all the nice software and data management and building out these databases um, prospectively, right? It's a lot easier to have a working database and these things in place than try to retrofit or populate later on. And so that's been a lot of the core efforts in the lab, is to build these enabling technologies so that we can just plug and play all the way through. Um, and have all these amazing data at the end to do things with. Okay, so just to reinforce that point a little bit, what's nice about this is even though cryo-EM is expensive when you buy it, the upstream part becomes a lot cheaper, right? So immune sera is usually accessible. And there's lots of it, lots of people doing different things. So you can get immune sera relatively cheap, and then you're not making a ton of antibodies. You're not doing expensive B cell sorting and sequencing and so on and so forth. So that gives you cheap inputs relatively, and then you can also avoid bias because most of the things that are published or most of the things that people work on are functional antibodies, whereas we look at both the binding and the functional antibodies, which will be really I think informative for antibody design and epitope prediction, so on and so forth. Um, yeah, so aspirationally, with these three microscopes propped up, if we get to 4,500 structures per year, we're competing with the total number of antibody antigen structures that are in the PDB. And again, I think by diversifying and magnifying, it'll be really transformational, and not just in terms of structures, right? We're predicting variants, and I showed in James's case we had 5 different ones. So you can imagine per variant, we can start to get even more data, and then if we start to model based on the clonal expansions from NGS reads, you get massive amounts of data points, and you combine structure, sequence, things like affinity and energetics, and you really have like the full armamentarium of data that can be useful for training and tweaking antibodies. So we're sort of cognizant of capturing and making all that data integrated. And what's nice is um, the field has moved really fast, and a lot of the big stuff like AlphaFold 3 that of course we don't have the resources to compete with are done. And so we can spend a lot of time in the fine tuning our transfer learning space, which is relatively lightweight. It still requires hardware, but it's the new data that we're plugging in that's the useful, tweaking and fine tuning. And Brian Coleman has a nice example of this with called thermo-MPNN, and taking thermostability data into protein design. So we again are sitting at a nice place where the big stuff has been done by the well-resourced and infinitely computationally enabled, and we can kind of fill in this space here. All right, with that, hopefully everyone followed along. I know I covered a lot, but really it's been a fantastic 10 plus years of us, creating new technologies really to enable a lot of the work that's going on um, in the rational vaccine and antibody fields. We've been really fortunate to work with large consortia and cohorts, um, from HIV to flu to malaria and so on, that really bring the resources, bring the clinical samples, bring the expertise that intersect well with our high throughput structural biology pipeline. And it's also enabled me to kind of build a amazing and diverse team that some of them come in as technology specialists, computer scientists, some have a particular interest in the disease, and they all can kind of leverage different parts of the backbone. And that broadness actually allows a lot of pressure testing on a lot of the tech and software developments. And it builds, a really robust and lively environment. And so I'd like to acknowledge, the amazing team, many of which I didn't mention in the talk today. And then also, the collaborators, funders, philanthropic donors, and then, the unsung heroes of clinicians and volunteers that they're conducting clinical trials, translating, these basic discoveries, and then working with us and connecting with us and interested in the data we have to offer, and sort of the new looks we can provide into endpoints for clinical trials and how to iterate. So it's a pretty amazing ecosystem that we've been able to participate in. And with that, I'll thank you for your attention, and you can go to the Q&A.
Wonderful. Thank you so much, Andrew. That was very, very exciting. We have a bunch of questions, nice questions. We have broad questions, we have specific questions. So I'm going to start with a few broad questions, okay? So someone asked, at what point is easier to solve the structure in the first place instead of trying to get 10,000 structures to train an AI to predict undiscovered new epitopes?
I mean, I think we are they're parallel approaches, right? So if somebody says, here's 20 monoclonal antibodies, we say no problem, we queue it up and hopefully over a weekend we get, through half of those. So we're opportunistic and it's not that, like I mentioned though, a lot of the antibodies that get sent to us are the functional and down selected antibodies. So they're inherently biased for different reasons. And so, the MPEM gives you a much more unbiased and broad look. So we do them both is essentially and using the same infrastructure. It just is opportunistic.
Got it. And do you think that using MPAN on sample from a clinical study showing lack of efficacy could help understand the lack of efficacy and help redesign antigen?
Yeah, I mean that's exactly what we're trying to do. We're not a primary endpoint in many cases, but we are sort of an important endpoint for that design process, right? And so in the example I showed where 75% of participants responded, the question is in those 25 that we didn't see it, did they have a genetic, an allelic variant that didn't work? Was there something else in there? And are there opportunities to design, for that patient population? So we just want to provide as much information as possible, and failures are arguably more important to look at than successes.
Got it. And someone said, great talk. What is the clinical relevance of the generated protomers generated after passive immunization with MABs?
We don't know, right? It's a phenotype that we observe, and we know that it happens after multiple immunizations, even in human patients. We hypothesize that, you know, if that's happening in vivo, you're opening up a lot of non-functional antigenic, epitope or immunogenic epitopes, and that's likely not a good thing, especially given how rare on-target responses are for HIV, for example. So we and others are, working towards eliminating that type of phenotype based on that hypothesis.
Okay. If such antibodies leading to trimer disruption are non-neutralizing, does this mean that the trimer disruption has no effect on the virion in vivo, or do they bind mostly post-fusion epitopes and hence non-neutralizing?
Yeah, so again, this is sort of a correlation, but generally, if you see a lot of the protomer phenotype, the serum is non-neutralizing. So that suggests that those are non-functional antibodies. We've never done a challenge study. I don't think it would be justified necessarily, but that would be what you'd need to do to really confirm that those responses are not are not efficacious.
And someone asked, I'm wondering whether interface monomer generating antibodies are uniformly non-protective in vivo. Should we treat interface binding antibodies as off-target, or is it plausible that some could confer in vivo protection despite showing no in vitro neutralization?
So there are some examples from the literature from Michelle Nussenzweig's lab and actually recently from Rich Wyatt that bind the interface, cause it to fall apart, but do have a neutralizing phenotype. The question is if you, and maybe that's good, but you're still opening up a lot of antigenic space for subsequent immune responses. So in an immune therapy setting, maybe you could use that, but in an immunization setting, I think you'd be working against yourself unless you had something that was, 100% broad and potent down to, nanograms per mil. But I think we're open to the idea that they could be okay. And then in the case of influenza, there are these antibodies that bind inside the trimer head and cause the trimer to fall apart. Those James Crow's lab has shown that they can be protective, likely through a cellular mechanism, right? So influenza infected cells present hemagglutinin, and these antibodies that bind can recruit, ADCC or ADCP phenotypes, and could be protective.
Are there long prongs in antibodies to CD4 binding sites in HIV?
Are there long what?
Long prongs,
Prongs.
PRONGS.
Uh, Maybe that they're referring to long CDRH3, which I will say actually CD4 binding site has very short CDRH3s comparatively. And most of the best antibodies are targeting via a CDRH2 phenotype. There are Tau antibodies, which have ultra long CDR3s that bind and neutralize the CD4 binding site, but those are a bit exotic.
Okay. Are the models like Angelo and Workflow open source? What are the computational requirements to run those models?
Yeah, so Model Angelo is open source. It's um, available from ShoreShare's lab. Um, that's pretty lightweight. The workflow that we're developing, we're actually, hoping to publish it and make it, open source and implementable. Um, and really all it's doing is efficiently running a lot of the rely on or CryoSpark or other routines that you would do in the structural biology workflow. So you still need the resources to run those jobs. But workflow will sort of automate and standardize, the different classification steps and different structure steps. So hopefully that's going to be available, soonish. We're still again beta testing a lot of it internally to make sure it all works. But yeah, the goal is to get that out there.
Okay. Andrew, we have like a bunch of more questions. I'm going to choose four more and then we can finish. Okay.
Okay.
So for MPEN, how do you count for neutralizing antibodies that have low affinity? And similarly, neutralizing antibodies that are in low abundance in sera, do you worry you might miss these clones?
Yeah, so that's a great question. When we get it, I should just put a slide in now as a disclaimer, right? So we estimate that we can detect 100 nanomolar affinity or better. Now, of course, that's contextual because if it's one in a trillion and that then you need a trillion observations to see it. Our typical data sets are about 1 to 5 million immune complexes. And like I said, from about 1% of the data, if it's there, we can see it. Um, so, that's tens of thousands of particles of observations, generally speaking. Um, we have done a lot of biochemical tricks like cross-linking, or we do it where we do a depletion of all the strong binders and then recomplex with what would be the weak binders or what didn't might have gotten competed out. And in both of those cases, we see a little bit more, but we don't see a lot more. So we don't think we're missing too much. Um, and also, in large scale campaigns, for example, with Ali El for flu, right? He'll make 200 antibodies, monoclonals. We'll do MPEM and compare the monoclonals, and we typically see more than all those isolated monoclonals combined. So, to the level that we care about, I think we're within the sensitivity and detection range that's meaningful. And honestly, chasing those rare or, homeopathic responses is probably not worth the effort in the end.
Okay. Okay. Yeah. I think five questions were about that. So I think you answered five of them. What is the feasibility of engineering de novo paratopes to target specific epitopes such as influenza NA?
I mean, it's quite good, right? As I mentioned, I almost don't even talk about the protein design tools because it's so much evolved and using a combination, AlphaFold, MPNN, it's quite easy to create these. And the Baker Lab and King Lab have done a tremendous amount to create a lot of scaffolds to design or add to. So that's kind of the passive part. It really is, then you have to go, well, one, you have to do an in vitro characterization, but more importantly, then you have to go in vivo, right? And you really rely on the outputs, the sequences, the structures and things that resulted from that actual design. And we still don't understand, even though we can in vitro design something that binds or presents an epitope of interest and binds the antibodies we want, it may not work in vivo. So it's back to the drawing board. And so the sooner you know that or can understand what's happening, then the sooner you can throw out that design or modify it or do something else that you want to do.
Okay. Does the model angel work without detailed knowledge of the antigen or without the knowledge of the most protective epitope to narrow down on?
Well, like I said, Model Angelo was really built not for antibodies, but for, looking at, let's just say, cell lysate and solving structures, right? But you could then search if it was like let's say a yeast cell, you could search the yeast cell proteome, and in that logo plot, you're going to find, you know, the correct sequence, the ground truth, if you will be embedded in that logo plot somewhere. It's a lot more difficult for antibodies, as I alluded to, because there is no ground truth and there's technically infinite diversity. So you have to kind of combine some inference, some of the modern, ablang and other tools, as well as the hidden Markov models of probabilities to kind of down select. So it's kind of a work in progress. And James is really the world expert at this right now. And I'm sure as it gets out there and more people are using it, we'll get better at knowing what to look out for and where signal to noise kind of steers you away from the right answer, and where you can make some leaps. Like I said, like in the framework regions of antibodies, you don't even have to really spend much time there because they're invariant.
Got it. And the last question, have you tried analyzing secretory IgA from mucosal lavads, or are you focusing most on IgG from sera?
So right now, mostly on IG Simcere, but Lee, who actually developed the microfluidic device, that's one of her aims of her thesis that she's actively working on. And we have BAL and we have other, samples that we're keen to look at. The primary difficulty is abundance and concentration, right? In those samples, secretory IgAs is limited. So Lee is working to actually create mini proteins that bind IgA specifically that she can enrich and then use that microfluidic device to work at a very small scale and enrich. And we'd love to compare IgG versus IgA specificity, and then also look at different vaccine modalities, right? Does a mucosal immunization result
Yeah,
in more or less, does a lot of the CD3 ligands and the things that people are doing, we really hope to get there, certainly by the end of Lee's thesis in another year or two, but hopefully sooner.
that's exciting. Andrew, thank you so much. And I'm sorry for the other question we didn't have time to answer, but I don't know, feel free to reach out to Andrew specifically. Those were questions about the MPEN technique itself. So feel free to reach out. And thank you so much, everyone. Thank you, Andrew, very much. And thank you all. Have a nice day.
Yeah, my pleasure. Take care.
Bye bye.